Okay, I guess we're on time. I guess we can. Uh, hey everybody, welcome back. Hi, Dr. Morris. We're very happy to have you join us in our sixth annual Plant Science Symposium. I would like to start by giving a brief introduction of Dr. Morris. Dr. Geoffrey Morris is an associate professor of crop quantitative genomics at Colorado State University. His research group develops and implements new approaches to understand and improve crop adaptation towards climate change resilience. His methodologies include mapping of genes that underlie climate adaptation and develop new approaches to better understand this process. His focal crop is sorghum, and he and his team work directly with breeding programs in developing countries such as Senegal, Niger, Haiti, amongst others. He's passionate about international research and education, focusing on soil, water, crop management practices, nutrition, and stress tolerance for improving crops productivity. In the US, he also works to facilitate the development of better adapt sorghum varieties in Kansas, Texas, and South Carolina. So with no further ado, please, Dr. Morris, the floor is yours.
Thanks so much. I am really glad to be here, and I just wanted to start giving a little overview of what I'm going to do today. Since I'm at the very end, and you've probably heard a lot of science already, and I'm really passionate about introducing students to the opportunities to use the scientific method. I'm going to do a talk that's a little different today. I prepared a special talk just for this symposium. I'm going to show you how you can use an applied scientific method to help you in your career. And I will talk about my own lab's research, but I'll be kind of using it really to give illustrations or examples of how you can use an applied scientific method. So I'm going to start by talking about why. Why do you do what you want to do? So how to develop your vision and mission, and to convince you, yeah, you know, you can think about trying to change the world, make a better place if you want to. And if you really get the goal setting right, you can really do that. The next step is really going to be that goal setting of what you actually do to understand your next user's goals and to set your goals to be able to help your next users. And then the how. How do you do that? How do you achieve your goals? How do you help your next user's goals? I'm arguing it's really through the scientific method. The amazing thing about hypothesis testing is you can start with the wrong answer, and hypothesis testing will get you to the right answer to achieve your goals and then eventually see your vision come to fruition. So I want to start by saying, you know, some scientists really have saved the world. You know, if you're willing to round up, Norman Borlaug credited with you know, basically 1 billion people not going hungry because of the work that he and others did. A couple kind of big picture lessons from the successes in the past. It wasn't hard, it wasn't easy, and it wasn't quick, right? And it wasn't done alone. You know, it's important to idolize people like Borlaug, who had a great impact, but it's also important to remember there were so many people who were involved in making that success. He was just one. I just brought up some key names, but of course there's dozens of people who devoted their careers to making this success happen in, for instance, the Green Revolution. So I'm going to start by telling you my vision. This is what motivates me. I have the vision that science could provide food security and economic prosperity for the world's drought-prone regions. This is what I think of when the work gets hard, when I get frustrated, when I get tired. I come back to this idea that this could happen. Why do I focus on this? It's because there's about 2 billion people in these drought-prone regions, and about 1 billion of them are in pretty abject poverty. And we also know that climate change impacts are disproportionately already affecting these people, and are expected to do more of that in the future. And when I say this, you might feel reasonably enough like, isn't this too big for one person, right? And of course, yes, absolutely, you're right. It is too big for one person. But I want to say it's okay to start by saying what you care about, right? And what about specific responsibilities, right? Who's going to do this? What am I going to do? It's important to take on responsibility, but I'd say first, it's better just to start with saying what it is that motivates you, right? Let people know about that. See if you have a shared vision. So the mission of my program is to understand and improve crop adaptation to drought-prone environments. And so I do focus on this crop, sorghum. It's a food and forage and feed crop around the world in these drought-prone areas. And so how is that mission different from the vision? The difference is I am taking responsibility for this mission. I hope that I'll meet some of you again in 10 years or 20 years, and you'll say, you told me you were going to understand and improve crop adaptation to drought-prone environments. What have you done? What have you done for that, Jeff Morris? And I work every day to make sure that I have something to tell you in 5 or 10 or 20 years when you ask me that. So I'm taking responsibility for that. You might wonder, where are the specific goals, right? And those are coming, but we want to think through our motivation kind of step by step. So the particular story I'm going to tell you about today, coming out of my lab, is the evolutionary rescue of sorghum, which is a climate resilient crop after a really terrible aphid outbreak that occurred in the Americas in the last decade. So this paper is coming out in a couple of weeks in Science Advances. And the reason this was of interest, for a broader interdisciplinary audience is a couple of things. We showed that half a century of shared work that was necessary for this rescue of sorghum to happen after the outbreak. We traced that history. And we also showed how you could use an understanding of that evolutionary rescue in order to rapidly develop new technology that will hopefully prevent some future outbreaks. Okay, so this outbreak was of the sugarcane aphid on sorghum, and so there's a biotype shift similar to what we're seeing now in COVID with the new variants that led to a really widespread outbreak in the Americas starting in around 2013. And so in the commercial sorghum agriculture, what that led to was a massive increase of insecticide use, and so that really reduced profitability for the growers and of course reduced the sustainability of the system as well. And that led to a very rapid decline of acreage for this climate resilient crop. It meant that the crop was getting substituted for much more drought sensitive crops in many cases. In Haiti, it wasn't just an economic issue, it was a food security issue. Haiti, the poorest country in our hemisphere. It's in the Caribbean. It has some humid parts, but a lot of the agricultural regions are actually quite dry in the rain shadow of the mountains. And so sorghum is the most important, really climate resilient, locally produced staple cereal in Haiti, along with rice and maize. It's an important part of the diet. And from 2015 to 2017, the sorghum landrace has almost disappeared. This is a sorghum field that got completely wiped out in Haiti that I visited. This is one of the few plants I could find that was at all surviving. Most of the plants died. The farmers replanted with corn. So they had a corn field, but that corn, as we know, is more susceptible to drought. And so this is basically what was left of the sorghum in a lot of places. And so there was a massive reduction in 2 years of the sorghum production, basically down by over 100,000 tons in this small country. So what happens when there's a huge change in environment like this? So in the ecology and evolution literature, they have this concept called evolutionary rescue. So you have this rapid environmental change, and there's a couple possibilities afterwards. One is, of course, extinction. The population level could get so low that it gets past a critical point where it cannot recover. And the other possibility is evolutionary rescue. And that is really just genetic adaptation due either to new mutation or to standing variation, polymorphism that's already there somewhere in the population. And this is thankfully what occurred in the situation in Haiti. There's a wonderful, wonderful breeding program, very new, by the University of Quisqueya. And they had just started a new breeding program when the aphid hit, and they were applying very intense selection for the aphid, and with no insecticide on a very diverse program that they had just built. And almost the entire program was wiped out. There was just a handful of survivors, but they were able to use those survivors, intercross them, rebuild a whole new breeding program, and very rapidly, incredibly rapidly, develop a new SCA resistant variety. And because of that, the sorghum production rebounded very quickly. A really incredible success, and that's really what we wanted to study. Sorghum was rescued in the Americas in just a matter of a few years. On the commercial side, the commercial sorghum side, there was existing hybrids. A few of them were found to be at least somewhat resistant, and those were released. So our question was really, how did this rescue happen, and how can we leverage knowledge on this evolutionary rescue in order to do a couple of things? One is to accelerate the development of new resistant varieties, because only a tiny number of varieties kind of made it through that bottleneck, right? So there need to be, they weren't the best varieties in other ways. So there needs to be more work on developing new resistant varieties. And also, how do we use this information to prepare for the next aphid outbreak or future environmental change? Okay, so I want to shift gears now and talk a little bit more about deciding what you're going to do. And I told you my mission already, and as was said in the introduction, I'm not a plant breeder, right? So how in the world can I achieve my mission if I'm a geneticist and not a breeder? And then the way I do that, right, is working with breeding programs. We work with breeding programs around the world. These are the public programs we work with. We also work with a number of commercial programs in the US. And we have to make sure our discoveries are designed to make it into those breeding programs, or else the program really doesn't have much of a direct impact. The way we do that is through these trait technology packages. And so typically, if the breeding program we're working with doesn't have the adaptive trait allele, then we will make a trait donor. So taking that allele and introgressing it into a suitable donor line for that breeding program. We'll make a trait marker, and that'll be low cost and will be outsourced, easy to use. So these breeding programs don't all have to become experts in physiology or entomology or whatever the adaptive trait is related to. And then we'll make what I call a trait map. And so this is like the scientific models of like, how does this trait work? How do you need to use it in the breeding program? So to get to your mission, right, you need to figure out who your next users are and what their goals are. And so this was my mission. And so I spent a lot of time talking with the breeders in Haiti and the breeders in the US trying to understand what they needed. And so on the Haitian side, they had the resistance, but they're very worried about the next biotype shift, the type of thing we're seeing with COVID now, right? One shift, one variant after the other. So they want to develop varieties with more than one source of resistance. On the US side, their biggest challenge is they need to convert these great hybrids they already had in the pipeline into resistant hybrids. Oops. Okay, and so before we made our goals, we first did this extra step of understanding our next user's roadblocks. So what do I mean by that? Like, where in their goals are they getting stuck? So in Haiti, the issue was they can't select for a new gene when the old gene still works, right? They could do the phenotypic selection very effectively. They showed that, but they don't know whether they're just reinventing the wheel every time, right? With the existing gene. On the US side, it's a little different. The aphid pressure is really bad on farms, but it's very patchy. So phenotypic selection is very inconsistent and very expensive. So it's very difficult to convert the varieties, their hybrids with phenotypic selection. So our research goal now, with that understanding in mind, was to identify a cheap, accurate, and global SEA-resistant marker that these breeding programs could use to select for, or depending on what they're doing, select against known resistance. So we developed and tested an SEA marker, and I'm not going to tell you how we did it quite yet. I'll come later. But we developed a CAS marker because these are low cost and outsourced, and these small breeding programs like in Haiti or mom and pop seed companies in the US don't need to develop a marker lab. And we tested these in an independent breeding population, or really just family that we had developed crossing US and Senegalese lines. And we used a greenhouse assay to test for the SEA response. And so here's one of the resistant progeny on the left, and the susceptible parent getting completely demolished by the aphid. And so, we genotyped these lines that we were testing, so we could classify them into homozygous for the susceptible associated allele, homozygous for the resistance associated allele. And you can see there's an almost perfect correspondence then between the phenotypes, which are there in this histogram on the x-axis, and the prediction based on the marker. So that gave us some confidence we were on the right track. But remember, our goal was to make a marker that was effective globally across different public programs and different private programs. And if you know the history of marker-assisted selection, there has been incredible success developing markers for individual programs, and a lot of disappointment developing markers, especially in the public sector, that were meant to be used broadly, right? In most cases, they didn't end up working for a lot of traits. So we also tested them in these markers in 4 commercial seed company programs, US seed companies, and 3 US public programs, which are kept anonymous since there's confidential information involved. And overall, we saw usually around an 80% match between the phenotype and the as determined by the breeder's own ratings to the markers. And so there was mismatches, which are important, right? Because this is what has doomed some of these efforts to make global markers before. So we know one issue is definitely that there's inconsistencies in the public germplasm, because we used public germplasm as for a lot of these checks. And we know that now over the last, 10 or 30 years since some of these public lines have been developed, things have got mixed up across the community, and that's always comes up whenever whenever there's new marker work. You always reveal what's been going on with the germplasm. We know there's phenotyping issues. That's why these programs, came to us or were willing to partner with us, because the SCA ratings in the field are very inconsistent. The other possibility is that there's complexity to the haplotypes. Maybe it's the same gene involved, but the haplotype that we're tagging with the marker is different, or there can be other genes. So there's a lot of potential hypotheses to explain what's going on, but overall, we were able to show that these markers were predictive. And because of that, then we were able to meet our next user's goals. So now this marker, which costs $2 all in to get per sample to get the genotype, it's being used by public programs in Haiti and the US, and now we're working with a program in South America. And one of the US seed companies, for instance, in the first year has ran more than 10,000 samples with this markers. So we're at a point now where we can say, yes, we've helped our next user achieve this goal, and pretty quickly too. So what I haven't told you, but maybe you're wondering, is how did we develop the SEA resistance marker? And how did we know, or why did we suspect the SEA marker would work globally? And that's kind of the fun part of this study, because it really brings in a lot of evolutionary and population genetics foundations into this marker development work. And the whole thing was really done with this goal-directed, hypothesis-driven research framework that we're using. That was one of the reasons why we were able to do this so fast. So how does hypothesis testing work? How does it get you to the right answer? So, I told you the research goal, but I didn't tell you about the hypothesis underneath it. This is the research goal I mentioned before. And so the first key point on this hypothesis testing issue is, that I'd like to convince you of, is that underneath every goal, whether you say it or not, underneath every goal, there is a hypothesis. And you can choose to assume the hypothesis, basically, and not state it, but it's still there. So why am I saying that? It's because, if you ask yourself this question, why do I think this goal is realistic? Why do I think this thing can be done? You have to think that there's some reality out there in the world that makes it so that goal is realistic. Something has to be true for the goal to be realistic, and that's what the hypothesis is. That's how you can find the hypothesis, is ask yourself, what needs to be true for this goal to be possible? That's your hypothesis. So for us, when we were developing the hypotheses, we did what everyone does, what you're supposed to do, dive into the literature to try to understand what was already known. In the aphid resistance, sugarcane aphid resistance case, there was one Chinese group that several years before had identified aphid resistant line, sugarcane aphid resistant line here, HN16, had crossed it with a US susceptible, and had mapped this RMES1 locus on chromosome 6. And so that's interesting, right? And the natural first question is, does RMES1 exist in other germplasm? Might it underlie resistance that we're seeing elsewhere in the world? But unfortunately, this HN16, there's nothing known about the origin, not available outside China. It's not easy to exchange germplasm with China. So on its own, it could be kind of a dead end. But this is where the evolutionary and population genomics kind of came in and unlocked the power of the work that the Chinese group had done. So, so we have this research goal, and then we need to kind of articulate the hypothesis, or a hypothesis that would make this research goal be tractable, right? So one such hypothesis is that SEA resistance in Haiti and in the US is due to RMES1. If that was true, right, then we should be able to track down alleles in the Americas that tag that RMES1 variation and make the markers. So the question, though, is how do we test this hypothesis? We don't want to just assume this and rush forward on a lot of marker development without knowing whether this underlying premise is true. Because that's why, one of the reasons maybe why some of the public sector marker development in the past has underperformed. So in this case, we could leverage this this set of concepts from evolutionary genomics, and that is that selection reduces genomic diversity, specifically in the loci under selection, for instance, by the breeders. So we did high density genotyping of the Haitian breeding program, several hundred lines that were developed after the sugarcane aphid selection, and we compared that to the genome-wide diversity for global sorghum. So in this plot, on the x-axis, we have the 10 sorghum chromosomes. Each position on the x-axis is a position in the sorghum genome. And then on the y-axis, we have a measure of nucleotide diversity, SNP diversity, so pi. So higher is more diversity, lower is less diversity. And you can see that amazingly, the Haitian breeding program still has a ton of diversity, only about a 50% reduction in diversity relative to the entire species, which is pretty amazing. And that was really because of the design of the breeding program to be very diverse. But if you look closely, you can see that there's regions in the genome that have lost a huge amount of diversity. And that is, based on theory, that's where we think the breeder's selection was applied. And so we've noted a few genes where we thought a priori, based on the breeding targets for this program, there was probably selection, for instance, against tannins or for stagrine. And so these signatures give us a way to identify potentially the signatures of the aphid adaptation. So there's two signatures. There's low diversity, and that's what I'm showing here. And then there's high divergence, which is the other signature of positive selection. And that's what we looked at next. So notably, RMES1 is at one of those spots where there's very low diversity. So can we fine tune this with the signature of divergence? So in this case, we were using fixation index, a very old evolutionary genetics concept going back to Sewell Wright 100 years ago. And it's just this classic measure of divergence. So we calculated FST for Haiti versus the global panel for each SNP across the genome, hundreds of thousands of SNPs. And under the hypothesis that RMES1 underlies SCA resistance in Haiti, then there should be significant FST outliers at RMES1. And so I'm going to show you how it turned out. Again, we have the 10 sorghum chromosomes on the x-axis. Now on the y-axis, what we have is the significance of the FST. So when this value is high, that means you have high divergence, which is what we would expect under selection. Okay, and so this is exactly what we expect. This is exactly what we would expect under the RMES1 hypothesis. RMES1 is found at the genome-wide peak for selection. It appears to be the locus under greatest selection in the Haitian breeding program. And that's true when we zoom in as well. This in red is the original interval identified with linkage mapping by the group in China. And here is the peak FST, highest divergence, highest FST SNPs right here. Okay, so, but we as a program that supports breeding programs, we need to understand this in the context of the breeding programs as they exist if we're going to make a marker that serves the needs across many public programs. So I spent a lot of time digging through like old pedigree records from like the 60s and 70s and 80s to understand where the germplasm people were using, where it came from. And was there a connection between the work, the aphid resistance in Haiti and the aphid resistance in the US and elsewhere? And so it turned out there was something that looked like a connection. This TX 2783, this was the major public SCA resistance line that was identified, and others with this SC110 in the pedigree. And it turned out that had the same progenitor as a West African SCA resistant variety, which was itself a progenitor of the Haitian breeding population. So we had a connection. In purple, we have the SCA resistant parents, and in gray, the susceptible. And so then the hypothesis is that based on this pedigree, there was a shared resistance allele, which could be the basis of a shared marker. a global marker. Let's see. Okay, so we also could make observations about the genome-wide kind of genetic similarity and structure of these populations. And we saw that the Haitian breeding program was basically nested within Ethiopian diversity primarily, whether we look at it with a principal components analysis on the SNP diversity or a neighbor joining tree. It showed pretty clearly that the vast majority of the diversity represented in the Haitian breeding program was diversity that came from Ethiopia. The breeding program knew that they used a lot of Ethiopian diversity, but they also used other global diversity. And so with this information, then we can develop a hypothesis really rigorously on the origin of RMES1. One thing to point out, to remember, scientific method is really about excluding hypotheses, right? It's not about proving hypotheses, it's about excluding them. So how do you develop hypotheses that you can exclude? One, you need to have two hypotheses, right? Because you're going to exclude at least one. So you always have two competing hypotheses. And then you take existing theory and previous observations and you make new hypotheses with that. So in this case, we use the kind of established theory in the sorghum world that SDA resistance is not common, been tested many times over many decades, with the observation that the Haiti lines cluster with the Ethiopian lines. Then we can make the hypothesis that RMES1 in Haiti is a rare variant from Ethiopia. And then contrasting to that hypothesis, we can take different theory and different observations and make a competing hypothesis that RMES1 is not a rare variant from Ethiopia. Maybe it's really just a Chinese thing. Maybe it's actually spread all over Africa. We know that SDA exists across Africa, so maybe there's been selection for it all over Africa. We know that the resistance was, association was originally found in a Chinese line, so maybe it doesn't have anything to do with Ethiopia. So now we have our competing hypotheses. So the next thing we need to do to exclude one of these hypotheses is to make predictions. Okay? The only way to really rigorously exclude a hypothesis, use the scientific method, is with a priori predictions. So these are the same hypotheses as before, and now we're going down to making a priori predictions about what we should see in our data if that hypothesis is true. So in the case where we, under the hypothesis that RMES1 is a rare variant from Ethiopia, then when we look at global diversity of sorghum land races, the resistance-associated SNP allele should really only be found in Ethiopia, right? By contrast, if that first hypothesis is true and the competing hypothesis, if the first hypothesis is false and the competing hypothesis is true, RMES1 is not a rare variant from Ethiopia, then there will be resistant associated SNP alleles, say, all over Africa, or not in Ethiopia, or only in China, something else, right? And we can visualize these predictions. Here I've kind of mocked visual predictions up after the fact using and modifying the real data. But under the for instance, under this second hypothesis, we would predict these resistance-associated alleles noted in red. We would, predict that they will be outside Ethiopia, right? Okay. So then, what did we see? We saw, and so now here we're looking at the real data. The susceptibility associated allele was almost fixed in the U.S. Just a few of those resistant lines were there in the U.S. In Haiti, as we already knew, the resistant associated allele was almost fixed. That's how we found this particular SNP here, which is the associated SNP. Okay, so that makes sense. Now, what about in the rest of the world? How did it look? So if you notice, the resistant alleles are only in this one corner in Africa when we're looking at global sorghum landrace diversity. And that little corner is, of course, Ethiopia. Right? So this allele is really only found in Ethiopia. And so then that allows us to exclude one hypothesis and at least tentatively accept another, and that is that RMES1 is globally rare but locally common variant from Ethiopia. And so just to kind of really show kind of rigorously what I mean by excluding of hypotheses using these predictions, right? So just replotting what you saw already before. So our observations matched the prediction under this first hypothesis and definitely did not match the hypothesis. Did not match the prediction under the second hypothesis. And so that means we can exclude the second hypothesis, right? Now, one thing's important to say, we didn't prove the first hypothesis, right? Because you can't really prove anything conclusively with the scientific method. What you can do is exclude hypotheses and see what hypothesis keeps surviving, experiment after experiment, prediction after prediction, right? And so we haven't cloned this gene yet, right? And so there's still the possibility that we will, when this gene gets cloned, we'll find out that there's something else going on. And so that this hypothesis was wrong, but this hypothesis lives to fight another day, and we'll keep testing it as rigorously as we can. Okay, so overall, what did we find in this story? This rapid evolutionary rescue, incredibly fast, just a couple years. It seemed really fast, but it took 50 years or more of global germplasm and knowledge sharing. So going back to that point, it wasn't quick or easy as it looked, and nobody did it alone. I'll just take you a whirlwind tour of the world for the path of RMES1. So, and then it'll be color coded by basically kind of like the decade that it happened in. So we know we have pretty good evidence that the allele originated from Ethiopia, right? Hundreds of years ago, thousands of years ago, we don't know when, but we know that in the 50s and then in the 70s, the Ethiopian gene bank shared it with the global gene banks, shared this one key accession, and that accession got over to African breeding programs, nothing to do with aphid resistance, just because it was a nice landrace that they wanted to use for breeding. And then through the 80s, 90s, and 2000s, the allele got shared around by breeding programs in West Africa and then back over to the US. And then in the 2000s, the resistance sources in the US germplasm were identified, but not in the US, because the aphid wasn't an issue in the US at that time. It was actually in Southern Africa, that the aphid resistance was actually found, and that information was sent back to the US, and as well as being used in Southern Africa. Then there was the discovery of RMES1 in China. And then finally, most recently, these collaborations between breeders and geneticists in US and Haiti and Senegal that allowed us to put this all together. So not quick or easy, and definitely nobody doing this alone, but the breeders, the gene bank scientists, the entomologists that were working on this really did save sorghum through this half century of work. I love this story because it really highlights the connections between all of us around the world, right? We have this one allele that connects African smallholder farmers with Haitian smallholder farmers and US commercial growers. And obviously it also connects those of us who consume this as a grain or consume the animal products that are fed on the sorghum grain. We're all connected, and the development of this marker really brings that home. Okay, so what have I done today in this talk? I've tried to give you a snapshot of this logic chain that we use in my group. I teach my students. I set this training material up and the framework up, but really what I was doing, I was reverse engineering how the best breeders, the best the best crop scientists that I worked with, how they thought. Okay, so Gael Pressoir, the breeder in Haiti, is an incredible visionary who sets goals and makes it happen. So like this framework is basically a reverse engineered version of how somebody like him thinks, or the wonderful wheat breeder at Kansas State University that I worked with, or many breeders that I met in industry. So breaking it down from the top to the bottom, why do we do it this way? And what's the what's the rationale for this? We need to first understand our own motivation, right? If we're going to be successful, if we're going to be a good collaborator. And so this question of why you do what you want to do, what I want to encourage you to do is take the time that you need to understand and to explain your own motivations. That's what I'm suggesting. And the flip side of that is don't rush into trying to define specific objectives. I started my lab, everything was GWAS, right? Because that's what I know how to do. That was my hammer and I was looking for the nail. Now I take much more time to understand why I'm doing what I'm doing before I rush into picking a particular approach and then trying to fit it in somehow. What you do. That is the key, is learning your next user's perspective. This is something I definitely learned from my industry collaborators, right? They're so good at that in industry and being focused on what their customer wants. We can definitely use more of that thinking in the public sector. So learn what your next user's goals are and why they're trying to understand why they're getting stuck. Then you can design your research to address the roadblocks, right? That same idea of just not rushing into exactly what you're going to do. And then the last piece, how you're going to get there. What I'm encouraging is really committing to the scientific method, taking that time to discuss your hypotheses with your colleagues, write down your hypotheses and predictions. It is like really, really fun. It makes science so much more rewarding. And it makes it more effective, I believe. And I have templates and training material that I'll share with you at the end to help you do that. So this last thing, I heard this quote first from pearl millet breeder in Senegal that I work with, the quote from Borlaug on that the first essential component of social justice is adequate food for all. And these are pictures from Haiti, kind of really underlying that connection. These are two pictures from the same hill. The ruins on the left, this is the fort where the rebellion really started, where Haitians overthrew slavery. On the same hill, just turning around, we're looking down on a field. This is a seed production field for the new aphid-resistant variety that the Haitian breeding program developed. So with that, I'd like to thank the members of my lab that did a lot of the work that I talked about today, and also especially thank my collaborator in Haiti, Gael Pressoir. And if you're interested to learn more about how you could use goal-directed, hypothesis-driven science, you can go to GoHy.org. Thanks very much.
Thank you, Dr. Morris. I think that was an incredible presentation. It was very easy to understand all that you said, and it was, well, I want to say from my part, and I think everybody very motivating. So thank you a lot. We have here a couple of, quite a number of questions for you. So yeah. Here's one. Are there any other traits of interest that could be linked to the RMES1 marker derived from Ethiopian plants?
Yeah. Okay. So that's an interesting one. Let's see if I can answer that without skipping back to the slides. So yeah. So this was a really neat example. This question brings up how the surprising ways that actually understanding the genetics of the trait can help, because when I started talking with breeders about aphid resistance, do you need help? Some of them said, no, we got it. But, and, others were like, yeah, and actually it's really hard to phenotype. But one of the things with RMES1, it is actually like right beside one of the major flowering time genes in sorghum. And so one of the hypotheses we're testing with the Haitian program right now is they are a bit concerned that there is linkage between one of the flowering time alleles that they need or that they've in the past used, and the RMES1. And so that's one thing that we're looking to. But the more we learn about the genetic basis of the traits, the more we're going to be able to actually anticipate problems of linkage drag instead of just pushing forward the breeding for 10 years and then realizing the problem afterwards. So that's actually the focus of a follow-on project that we're doing with the Haitian program, is dealing with those linkage issues. It's a good question. I see I have the Q&A up, and I see from Jesse Murray again, perceive a lack of evolution-based concepts or analysis in breeding-focused research. That's such an interesting question. I say I see sometimes like too much and also too little. So my background is evolutionary genetics. I was really excited to try to apply evolutionary genetics to concepts to breeding. And I first started really working with crops as a postdoc research assistant professor in South Carolina with Steve Kresovich. And he was like, what everybody says about crop geneticists? All they do is come in after the breeders have done all the hard work, and let's just make a nice paper out of it. And that really stuck with me. And I think that perception is largely true, right? I don't think the breeders are that far off when they say that. That is mostly what geneticists in the public sector have done. And so this paper, I'm so proud of it because it's the first thing I've been able to hold up and say, here, evolutionary genomics made this tool. And this marker would not have happened because other people are working on markers. They just don't have them done. We did it faster because we were able to leverage, number one, a great breeding program. It would not have worked if the Haitian program wasn't so well designed. But we also leveraged evolutionary genomics. And so to my knowledge, this was the first example where we proved that evolutionary genomics was useful to breeding. Yeah, so I think there's a lot more evolutionary. I think there's a lot more that evolutionary geneticists could do to meet halfway. That's kind of my thought. Okay, what else?
We also have a couple questions that are coming here through the chat. Here's one. Do the aphids carry any other disease, or is the foraging the main issue with the aphid?
Yeah, so it's like, do they carry like viruses or something like that? I've not seen anything in the literature to suggest that that's the case, but it's a great hypothesis to consider. And I do know my colleagues down the hall who work on aphid virus interactions are keeping an eye out for any evidence that hypothesis might be true.
Yeah, well, that question was also related that it should be easier to discover well, let me just read it, please. It should be easier to maintain resistance to the insect pest than fungal or viral pests. But is it easier to discover it?
Easier to maintain resistance, and you're thinking that the biotype shifts are in the aphid are slower than the I'm blanking on the technical term for the shifts in the fungal or bacterial side, but basically you're saying the idea is that the arthropod pests evolve more slowly.
Yeah.
So I don't know. So there in sorghum, there's pretty good evidence that in the 80s and late 70s to the 80s and early 90s, basically all genetic gain stopped in sorghum because they had this green bug aphid problem. And there was, I think 3 or 4 major biotype shifts. So they happen really pretty quickly. So I think the idea that we might have to deal with biotype shifts, I think it's fairly likely, especially because it seems like most of the industry is using, or like most of the community is using probably one major gene. We do suspect that there's more. What was the second half of that question? Sorry, I forgot.
So was it easier to discover it?
Oh, yeah. The only reason this looked easy is because of how hard the Haitian program worked and how well designed that breeding program was. Those evolutionary genome scans, there's hundreds, thousands of papers, in crops using those evolutionary genome scans. Why haven't all those turned into markers before? It's because they weren't doing the genome scan on a basically perfectly designed breeding program for those genome scans.
We also have another question here. Can you talk a little about how you identified the SNPs associated with the resistance and developed the CASP markers?
Yeah, sure. So the main goal of this project is a project with the Gail Pressoir, the Haitian breeder, and Ed Buckler, Cornell, funded by USAID. And the main goal was to test whether genomic selection in a developing country breeding program was actually feasible. It turns out it is. We've been doing it now for several years. My lab's been helping, but the program in Haiti has been doing it for several years. So we had genotyping by sequencing data for the whole training population. So that was where the SNPs came from, was genotyping by sequencing. So fairly high density. We're not getting all the variants though, right? And so then how do we identify it? It was really just completely off the shelf genomic selection, or sorry, not genomic selection, genomic signatures of selection tools. In the world I came from, FST, the old test, nobody would use that. You'd be embarrassed to try to publish a paper with that because there's much cooler things. But I picked that on purpose because if any of you ask me, like, how does FST work? I can actually write it down. I can write down the equation. The new methods, even as a professor of quantitative genomics, don't understand all of them. But we just took the simplest method and we set up our hypotheses well. We built off a really great breeding program. And then we got a very clear answer. It might have gone the other way, right? We might have seen no signature of selection on RMES1. If that had occurred, we would have had very, quite conclusive evidence that it was a different gene, right? So either way it turned out, it would have been useful. It would have been interesting.
And I think we have maybe time for one last question. I have here, what was the behavior of the heterogous zygous plants for the resistance genes? Do you know
yes.
information about that?
Yeah, okay. So we did all our studies with homozygotes, in part because we had the markers, so we could find the homozygotes. The training population in Haiti they were all inbred at the point that we were studying them, but it was already known in the literature the resistance is dominant. So actually, a really nice control that maybe we should have done in that first validation that I showed you, that kind of U-shaped curve. A nice extra control would have been to make sure that the hets have the right aphid resistance reaction as expected. Actually, the reason we did that is because we didn't do that is, we needed to use, I think our entomology colleague used maybe 6 plants for the ratings. You needed multiple plants. That's actually why we couldn't use hets. But yeah, it should be dominant.
Okay, Dr. Morris, thank you again for the beautiful presentation and excellent explanations of everything. I think everything was very clear. And now with this, we conclude our presentations for the Plant Symposium. I want to thank all of the speakers also for their incredible presentations, and I would like that our attendees also thank them for logging in. It means a lot for all the Plant Science Council at the University of Florida, and it's very important, all the knowledge that we've shared here.